Hello everyone, am I audible and visible? Let me know, then we'll start. Am I audible and visible? Okay, fine. So, hello everyone, I'm myself Dr. Deepthi Karya and I am your faculty for Physiology. Let us start today's topic. Today we will discuss about physiology of gastric secretion. Okay. Before that, I would like to inform you all that you can get soft copy of all my notes by downloading the application Vesalius, and you have to subscribe for the same. And second thing, that is, if you wish to get hard copy of all my notes, you can order either on the Flipkart or on the Amazon. Okay, and you can join my WhatsApp group. That is number is 78638 22668. Okay, you can join through link also. Link of all of this that is given in the description box. Okay, now let us start our today's topic, that is physiology of gastric secretion. Now, gastric secretion, that is the secretion of stomach. Okay, gastric secretion includes two things. One, that is secretion of the exocrine gland, and second is the secretion of endocrine gland. So exocrine, that is gastric juice, and endocrine, that is gastrin hormone. Okay, now let us discuss with the gastric juice secretion. You can see here the gastric juice consists of water 99.5% and solids 0.5%. Okay, this solid again convert, it consists of inorganic and organic substances. Okay, so if we are talking about inorganic, all the ions are there, mainly your hydrochloric acid, one of the important constituents. Other also you can write down sodium, calcium, potassium, bicarbonate, chloride, phosphate, sulfate and all. Organic substances include your enzymes, pepsin, renin. Renin is present only in the animals, not in the human being. Lipase and gelatinase. Okay, and urease is also present, and other organic substances, they are mucus and intrinsic factor. Now, what is the normal volume of the gastric juice? That is about 2 to 2.5 liter, 2 to 2.5 liters per day. Okay, or 2500 ml per day. pH is strongly acidic. Try to remember the pH 1 to 2. Okay, so this is about the general composition of gastric juice. Now, which are the enzymes present in the gastric juice? Very important enzyme, that is pepsin, which is your proteolytic enzyme, lysis of the protein, and this is secreted by peptic cells, which are also known as chief cells. Okay, chief cells or peptic cells, these are present in the gastric glands. Okay, this pepsin, that is in inactive form of pepsinogen, and this pepsinogen is converted into pepsin by HCl. Okay, another enzyme, that is gastric lipase. Lipase, that is lipid fat splitting enzyme, but this is weak enzyme, fat splitting weak enzyme, and it has little importance because this will not act at this pH. Okay, so it is useful only in case of when pancreatic lipase is not working. Okay, not sufficiently working. Next is gelatinase, that is to liquefy gelatin, that is present in the connective tissue, commonly in non-vegetarian diet. Amylase for carbohydrate, that is also in small amount. Lysozyme, this is the enzyme which destroys bacteria, so this is bactericidal enzyme. Okay, Raj, you are talking about trypsin, chymotrypsin. These all are present in the pancreatic juice. We are talking about gastric juice. Okay, this will also come in pancreatic juice. Fine, this is pepsin here. Fine, pepsin and amylase in small amount. Okay, carbonic anhydrase is also present. That is secreted by parietal cells. Okay, and urease, that hydrolyzes urea. Okay, and urea is converted into ammonia. Fine. So these are various enzymes. Another important Ali, hello, welcome to my class. Fine. Now be attentive. Now next important constituent of gastric juice, that is mucus. Very important thing, because if mucus is not there, then what happens? Your acid degrade this mucosa. Okay, two types of mucus are there. One is soluble mucus. This is secreted by mucus cells which are present in the pyloric and cardiac glands. Okay, and insoluble mucus, that is secreted by surface epithelial cells. Surface epithelial cells, which are known as foveolar cells. Okay, these are, hello Kishan, good evening beta. Okay, so this mucus is also secreted, and intrinsic factor, one of the very important constituents which is secreted by parietal cells. Now I repeat all the secretions from the gastric juice. We have discussed enzymes are there. Enzymes are pepsin, gastric lipase, gelatinase, amylase, lysozyme, carbonic anhydrase, urease. Okay, mucus, intrinsic factor, and HCl. Okay, that is secreted by parietal cells. Now we will discuss secretion of gastric juice. Now secretion of gastric juice means secretion of important constituent, that is HCl. Secretion of enough, good evening beta, good evening, welcome to my class. Then secretion of water, secretion of pepsinogen, mucus, gastrin, and intrinsic factor. We will discuss one by one. Okay, now starting with secretion of HCl. Okay, as I told you, easy way to remember parietal cells, you have to remember PIH. I means intrinsic factor, H means HCl. Parietal cells secrete these two things. Okay, easy way to remember PIH. Parietal cells, they secrete intrinsic factor and HCl. Fine. Now, so this is secreted by which cells? You can see here. These are parietal cells. They have canaliculi. You can see here canal inside. In this canal, you have HCl which is secreted. Okay. So these are, you can see, complex network of intracellular canaliculi which is present. Okay. Now another important thing, about 2.5 liter of HCl per day is secreted. And because of this high amount of HCl secretion, you can say that pH is highly acidic, 1 to 2, okay, of gastric juice. Now this, in stomach, this HCl is secreted in H plus and Cl minus. You can see this is the diagram which easily you can, with the help of which you can easily understand how HCl is secreted. Okay, so the concentration of H plus ion increases as H plus secretion increases. Okay, you can see, now we will discuss various steps how HCl is secreted. Now another important thing here is, when we are taking specimen of parietal cell secretion that has been obtained, that is having very high concentration of H plus ion, and the pH is very low. The pH actually, if suppose pure, if you are taking pure secretion from the parietal cell, the pH is 0.87 only. So how the pH becomes 1 to 2? Because we have food also. So the food neutralizes some of the acid, therefore the pH increases here. Okay. Okay. And inside the cell we have isotonic solution. Okay. So pH of the cytoplasm, that is equal to other cells, 7 to 7.2. So how the cells having normal pH, how can they secrete this much of acidic secretion? We will discuss the mechanism of secretion. There are various theories were proposed for the secretion of HCl. One is Davenport theory, Davenport and Fisher. If you can remember, then it is okay. And other is Davis and Hollander hypothesis. Okay. Now, so how are they secreted? H plus separately, Cl minus separately. Okay. And secretion steps. Now we will discuss the steps for the secretion. You can see here, first of all, inside the parietal cells, you can see here, first step is H2O combines with CO2 here inside the parietal cells, and in presence of carbonic anhydrase, these steps are very fast. So it forms H2CO3 first. You can see here H2CO3 here. Now this H2CO3 is immediately converted into H plus and HCO3 minus, again with the help of carbonic anhydrase. Now what happens here is the lumen. So this H plus ions, they are secreted inside the lumen, and this bicarbonate ions, they enter in the blood. Okay, so this is the basic step. Fine. Now another important thing, this H plus ion secretion is by active transport, H plus K plus ATPase pump, this one. Okay, and this H plus K plus pump, that is inhibited by the drug omeprazole, and this drug is given to reduce H plus ion secretion because the pump is inhibited. So you can say that now H plus ion cannot be secreted. Am I clear? So this is the applied aspect. You can see here, these are the steps we have discussed: CO2 plus H2O, H2CO3, H plus HCO3 minus, H plus ion and K plus, H plus K plus pump. This is inhibited by the drug omeprazole. So if you have acidity, you are taking the drug omase. Postprandial alkaline tide. We are coming to the point. Good. You have read the topic. Very good, Ali. We will be coming to the point. Fine. So this is the omeprazole. This is the applied aspect. Now next thing is, you can see here now what happens to this bicarbonate ion. This bicarbonate ions, they exit, they enter in the blood. Okay, they release in the blood and they are responsible for postprandial alkaline tide. Very good. Okay, so what happens, one side H plus are there in the gastric secretion, but blood becomes alkaline because of HCO3 minus, and this HCO3 minus transport, that is by counter transport, or that is known as antipod, in opposite direction. HCO3 minus ion moves out, chloride ion enters in. Fine. So when HCO3 minus ion release in the blood, that is known as postprandial alkaline tide, one of the very important viva question or short question. Okay, so what happens here, alkaline tide means blood and urine becomes alkaline, and because of alkalinity, respiration is also depressed, and therefore PCO2 increases in the alveoli because of depression of respiration. Okay, so this is all about postprandial alkaline tide. So whenever acid secretion, you can see here, increases here H plus K plus ATPase, along with that there is definitely increased absorption of bicarbonate, and that produces postprandial, means after food intake. Postprandial means after food intake, because after food intake we require to increase secretion of H plus ion. Okay, and therefore there is alkaline tide, urine is alkaline, blood is alkaline, there is slightly depression of the breathing, and because of that alveolar partial pressure of carbon dioxide increases. Okay, now another thing, now how this chloride ions enters, you can see here, this HCO3 minus ions, they enter in the blood, and chloride ions, okay, chloride ions are, they are transported by secondary active transport. Secondary active transport, this is enough, Ali. This much description is enough in the class, I think so. So secondary active transport. Okay, so they are transported here by secondary active transport, and along the gradient now they enter in the lumen. Okay, another important thing here, that is chloride channels are there for the transport. This chloride channels, they are activated by second messenger system, that is cyclic AMP. So you can say that H plus also enters and chloride ions also enters in the lumen, and there is secretion. This is, you can say, the secretion of HCl. Fine. Now here for this we require large amount of ATP, because H plus K plus ATPase as well as your HCO3 minus chloride antipods. So all these actions, they require ATP. So this ATP is from where do this ATPs they come? They come from the mitochondria. Here are the mitochondria. Okay, so this mitochondria, they produce ATP. Mitochondria present 40% of cell volume of the parietal cell. So large amount of mitochondria is there, and that is responsible for this ATP generation, which is required for all this transport. Okay, now another important thing is here there is negative potential in the canaliculi here. This is negative lumen. Okay, this is minus 42, minus 70. That is also important because that produces gradient. And why this becomes negative? Because of sodium potassium pumps. Okay, sodium potassium pump is located at the basolateral surface. This is apical surface, this one. This is basolateral surface. You can see here this one, sodium potassium pump. So what is the action of sodium potassium pump? It pumps 3 sodium out, 2 potassium in. So you can say that because one positive ion moves out, there is a gradient which is intracellular negativity which is created here. Okay, and this potassium ions which are entering here, this will be moving inside the lumen. Okay, through potassium channels. This is potassium channel. Okay, they are present at the basolateral as well as apical membrane. Okay, and this diffusion further increases intracellular negativity here inside. Fine. Now, certain factors they stimulate HCl secretion, and certain factors they inhibit HCl secretion. Very important. Try to remember this all. Vagus stimulation that releases acetylcholine, because your parasympathetic nerves they always increase all secretions. So vagus, that is parasympathetic, so it increases secretion. Then gastrin hormone also increases secretion, and histamine also increases secretions. Okay, so this all you can see here in this diagram: gastrin, acetylcholine, and histamine. We will discuss this thing in detail. How they increase secretion, we will discuss in detail along with peptic ulcer. Fine. Now, which are the factors they inhibit HCl secretion? When first factor, when your pH becomes highly acidic, large amount of acids are secreted in the stomach, and when the pH is very low, then now HCl secretion decreases. Second thing, certain intestinal hormones like your cholecystokinin, somatostatin, you can see here they are inhibitory to the parietal cells. Okay, so they inhibit the secretion. Some other factors like prostaglandins, epidermal growth factor, transforming growth factor, they all inhibit HCl secretion. But important, very important factor is low pH. Means when your when your gastric acid juice or gastric acid secretion increases, what happens to pH? pH becomes very low because of high H plus ion. And now when high H plus ion, now we need not require to get the secretion. So you can say that secretion is inhibited. This is negative feedback mechanism. Okay, so you can see here these are your acetylcholine increases. Okay, but somatostatin here it inhibits. You can see here acetylcholine and gastrin stimulate the secretion. Fine. Now, what are the functions of HCl? Very important function is for activation of pepsin. We require HCl. Pepsinogen is converted into pepsin by HCl, as well as secretin is also activated from prosecretin by HCl. Very important other function. This is first function. Second function, optimum pH for action of pepsin. Pepsin acts in acidic pH. So if the pH is alkaline, it will not work. Third, bactericidal action. Suppose if you have taken some unhygienic food, है ना, बाहर जाके पानी पूरी खा लेती है तो क्या होता है? Bacteria they enter. So these bacteria are destroyed by this acid. You can have you are surprised to know that this HCl can destroy blade also. Metal can be destroyed. This is so strong HCl in our stomach. So all the bacteria are destroyed. Fine. Then iron absorption also requires this acid. Okay, because iron is placed in Fe plus to form in the presence of this acids. Bile and pancreatic secretion are also stimulated by the acid. Okay, I repeat, for activation of pepsinogen, prosecretin, HCl is required. Second, They provide pH for the action of pepsin, destroys bacteria, iron absorption, bile and pancreatic secretion are also stimulated. Next is we have pepsinogen. Pepsinogen is inactive form and it is secreted by peptic cells, or other name for the peptic cell is chief cells. Okay, secreted by chief cells. This pepsinogen that is secreted in small amount by pyloric glands also. Mainly these cells are present in the glands of the body. Okay, pepsinogen is secreted by chief cells. Okay, they are pepsinogen 1 and 2 and that is converted into pepsin by HCl. So this is stored in the zymogen granules. You can see here chief cells. You can see here these are the granules. They have pepsinogen. Okay, and this zymogen granules they release pepsinogen and the secretion is increased by vagal stimulation, gastrin and histamine. And it gets activated by HCl. Number one. Second thing, pepsin. Pepsin also has autocatalytic action. Means pepsin activate other pepsinogen. Means small amount of pepsin, once it gets activated, it activates the remaining pepsinogen which was inactive. Okay, that is another action. Now what are the functions of pepsinogen? It is a proteolytic enzyme. So what does it do? It converts, it splits proteins into, so suppose this is a protein, long chain amino acid, so it cuts it, so it is converted into proteases, peptones and polypeptides. pH required for the action of pepsin is highly acidic, minimum 2 pH is required and that is provided by HCl in the stomach. Okay. Next secretion, that is mucus, as we have discussed insoluble and soluble mucus. I am not going in detail. Insoluble mucus is secreted by mucus snake cell and this is by a foveolar cell. Okay. This is same we have discussed. I am not going in detail. Now another is intrinsic factor. Intrinsic factor, as I told you, PIH. Parietal cells they secrete intrinsic factor and HCl. So intrinsic factor is also secreted by your parietal cells. Here you can see parietal cells. So this is a glycoprotein secreted by parietal cells and this secretion is linked with the action of histamine and the secretion is by cyclic AMP mechanism. Okay, cyclic AMP mechanism. Now what are the important functions of intrinsic factor? It is required for the absorption of vitamin B12. How? You can see here in the stomach you have, this is suppose intrinsic factor and this is your vitamin B12. So intrinsic factor binds with the vitamin B12 here and it protects vitamin B12. It will not allow the vitamin B12 to be destroyed by gastric acid. So now this vitamin B12 intrinsic factor, it will pass riches to the ileum and in the ileum B12 is absorbed and your intrinsic factor will go back. So it forms a complex with vitamin B12 and it prevents its degradation in the stomach. Okay, so if there is vitamin B12 deficiency, most of the time it is the deficiency of intrinsic factor because intrinsic factor is required for the absorption of vitamin B12. So vitamin B12 deficiency, always we are giving injections, not the oral way. Why? Because if we are giving orally but intrinsic factor is not there, then it will not be absorbed. Fine. And deficiency of intrinsic factor and vitamin B12 produces anemia, which is known as pernicious anemia, in which RBCs are megaloblastic, large RBCs with the nucleus. We will discuss in detail with the pernicious anemia and megaloblastic anemia. Okay, so this is all about gastric secretion and different constituents and their functions. Fine. So again, if you wish to get all my notes, you can download the application and subscribe. The application is Vesalius and my notes is also available on Flipkart and Amazon, that is Physiology Notes. And if you have any doubt, I can give you my number, WhatsApp number. You can ask me, you can direct message me regarding your doubts of physiology. My WhatsApp number is 78638 22668. Okay. and you can join also my WhatsApp group. Link of all that is given in the description box. Fine. So thank you for being with me. If you like my video, you can like it, you can share with all your friends, and you can subscribe my channel. Thank you so much.